I will walk you through a voxel-based morphometry VBM analysis. This is going to use SPM12 and CAT12. We have previously used CAT12 in a previous module, and so this will just build on that. And this will allow us to compare two groups of individuals, look at their brains to see if they have different amount of gray matter in parts of their brains. So the data for this are in module 7 here. This is our share drive under data, and you'll copy this directory over to your own directory. Again, don't do any work in here. You can copy it this way. You can navigate to the blue Sci 4930 share data location, check the box by module 7, hit copy, and then this will show up. Don't push any of these buttons yet. You want to navigate to the location where your data are. And there's different ways to do that. You can just kind of click around. So like this, I'm going to go there. And I have a folder here in share. You're likely are going to go to share students and then into your folder, or maybe blue Sci 4930 and then your folder. Again, don't put it in my folder, put it there. And so then I'm going to copy it this way. So I went to my folder, and then there is the copy of this. So we can click in and see we have a group of brains from control participants and a group of brains from people who have schizophrenia. And we can click in and see there are the nifty files within each of the folders. Okay, so I have a copy of the data. I will show you one more way to copy it, but once we get the MATLAB open. So this will use MATLAB, and I've already requested a session. But if you log in, do MATLAB, I recommend 2024B or an older version. So this might default to 2025B. I've actually had some issues with 2025B on this processing. You can try it, but it gave me some problems. And so I'm going to do 2024B. 8 cores, 32 gigabytes of RAM. And then make sure under the Slurm account in the QoS you have, so the class Sci 4930 and QoS request burst access. And that's going to be important because we don't have an unlimited number of cores available, and this will allow us to have more temporarily to do this. And then request 24 hours of time. This won't take that long, but the processing can take 6 hours or more. So keep that in mind. And you'll click launch. I've already done this, so I will launch MATLAB. Okay, so this is 2024B, the version. And first thing we want to do is we need to make sure SPM is available. And so to go there, I will open the terminal. So Applications, XFCE terminal. And then I will cd blue Sci 4930. And that will force a connection to our class drive. And then over in the MATLAB window, we can now set the path. And so there's two ways to do it. You can just click set path and navigate to it, or you can type in the path here within the directory. So right here, you can do blue Sci 4930 share data like that. And we can see here's module 7, NeuroTools, all of this. So sometimes I find it easier to put the path in there first, and then when I go to set path, when I do add folder, I'm already just about at the right location. So I can just click on NeuroTools, that's a double click, and then single click on SPM12, hit open, and then hit save. So I got this message, MATLAB cannot save changes to the path. You can save it to a different location. Do you want to save to a different location? So yes, and then this comes up. So it's going to be important to save this to somewhere you can write to. You have two options. You can save it within your folder. Again, don't save it into a general class drive, only do it in your folder. And so it could go in Sci 4930 and then in your folder within that. Again, you need to make sure you're within the location, or it can go in Share, Students, your folder, or you can save it to your home directory. And so we can click on the little home there. And this is in a different location. This is not on Blue Sci 4930, but it's okay to save this path there. So I'm going to click Save. It said, I already have one. Do you want to replace it? Yes. And you should not get an error if you do that. So saving it in your home directory is really the simplest place to put it, but it said hopefully not an error, and then hit close. I'll say if you use MATLAB 2025B, you do not have to save the path. You can add it and it will remember it for that session. But again, I had some issues running with 2025B. So close that. And now the test is, do we have SPM available? So within the command window of MATLAB, type in SPM. And it's loading. It didn't give me an error. So here this window is popping up. Before we go through the process, I just want to show you one other way to copy the files if you haven't copied the files already. So you can also copy the files using the terminal. So I could do it within here. I could do the terminal over on the HyperGator desktop session. So again, that can be opening something like a shell access. And this might take some more seconds to load, but you can wait for that to load. This will be a terminal window, and you can copy it that way. But I'm going to do it. I won't copy this. I'm just going to show you how to do it within this side. So remember, we had data within our class drive. So Blue Sci 4930 share data within that location. So I'm going to go into that location. So I'm already in Blue Sci 4930. So now I can just type cd space share / this / data. Type ls, and we have the module 7 directory there. So we can copy this. So cp space -sign is what it is. Just what we call the dash is the same thing as -sign on here. So dash lowercase f, which is going to force this to do it. I do a capital R, which is recursive, and then space, and then you can do module 7. So again, within this drive, within the Blue Sci 4930 share data location, and then I want to copy that to Blue Sci 4930, and then you might copy it to your folder within there, or your folder within share students, kind of that location. And so I have a folder in share. Mine's not in students, so I have a folder in there. And what this would do is copy this module 7 into the directory that you are typing there. And so that's my path. Yours is going to be different. And then the one thing to make sure is on the end of this, you have a /, and then that way it'll make sure that you're copying this folder over into that one. So then you put enter return. It would be a quick process, and then you're all done. So I'm not going to do that because I already moved the data over otherwise, but that's the command to move data over. And then once that's done, you can close the window. So now to run this, we click pet and VBM. So we had done this before when we were doing the module 4 assignment. This will just take about that long, 10, 15 seconds to load up. We're not going to use SPM right now. We're going to use CAT12. So we can come over here and click toolbox, and there should be a CAT12 toolbox. If there isn't, then you likely added a different SPM12 folder. And then, like we had done before, we click segment. So on the Module 4 assignment, so this is the first step for doing the voxel-based morphometry. So now we just need to load in the volumes. So our volumes are going to be all of those ones that we copied over. So I can double click on that, and then notice that I'm in the incorrect location here. I'm in this share data location. I need to get to share and then get into my directory. Sorry, that was quick. After that initial double click, these are all single clicks. And so I'm in now the correct location. For me, you need to make sure you're in a different location where your data are. So then within Module 7, VBM. And there's the 2 folders in there. Now the easiest way to do this is just to be in this location, and let me make this window a little bit larger. So over on the right side, before this dot and then the little asterisk, I'm going to type NII, and so this should say NII dot star. And then I can come over here to the left and click the REC button. This is going to recursively select files with current filter. And if you've copied over all the data correctly, then this should add 40 files. So we're doing I just want to mention one thing. With this type of analysis work, we are comparing 2 groups of data. So we have a group of control participants and a group of people who have schizophrenia in their brains. And we want to make sure that when we're doing this type of analysis that the groups are grouped together. And this is why we have 2 separate directories, our controls and our patient group. And I always like to have the control group come first alphabetically. So then that way they are always kind of the baseline reference group. You don't have to do that, but just kind of if you're setting this up for your own research or with the research for a kind of faculty member, professor, that this is going to be kind of an organized way to do it. And we can verify that all the controls are added first. We scroll down, see all the con. We have 20 of those, and all schizophrenia, and there's 20 of those as well. So if you don't see selected 40 files, then something was wrong. You need to try this again, kind of go back, and or maybe you didn't, all the files didn't copy over, but you should have 40 files. So at that point now, I'm going to click Done, and we should see a list over here, all the 40 files, and it says 40 all of there. Now, we don't have to do anything else other than click the Run button. So this will take some time to do. So I will click Run, stop the video, and we will come back when this is done. So we can see in the MATLAB window, it's running job 1. And here's the log file that'll pop up. It'll start the next job. It'll have another log file open. So this is a way that you can check to know when it's done. But just give this some time. This can be some, you could check it in an hour from now and see if it's done. And if it's not done, then, and it'll tell you if it's done. You won't get an email or anything, but you have to look at the window and it'll tell you if it's done. If it's not done, then come back half an hour or an hour later, and once it's all done, then you can finish everything up. Okay, I'm back to show that the processing has finished, although we will see that it really hasn't all finished. So within the main MATLAB window, I went back to my window, I'd closed it and I opened it back up. Again, just kind of like this process. Over my interactive sessions, launch MATLAB, load it up. We see right here, completed jobs, 1 out of 40, all the way down to 27 out of 40, and then it stops. And then otherwise it says conclusion of 40 cases, some process with warning, we'll ignore that for now, and then 13 are unknown or unprocessed. And that's what we can see. There's actually some brains that didn't fully process. So we need to reprocess those, and sometimes this will take a couple of steps. So let me show you what we're expecting. So I'm in the VBM folder that I had copied over, and I'm doing this processing in. And if we look in this list, we see con subject 1 looks like they completed. So I go to con subject 1, a NAT, and then look in. We have a lot of files in here. And so about 27 different files in here, and that's about what we should have if it completed correctly. But importantly, we should have a CAT report, and this is what you had submitted for the module 4. So if you open it and look at it, it should have some information about the scan, have the pictures, some data, how much time it took to process, all of that. And if this file exists, then that means the processing completed. If it doesn't exist, something happened, it didn't process correctly and completely. So that exists for 1. If we look in the list, we see there's a gap between 1 and 5, but we do have a subject 2. So 2 didn't finish. We only have 9 files in their directory, and it should have a lot more than that. We don't have that CAT report. And so it just didn't finish. And so what we can do is we can find that CAT report. So there are kind of different ways to do it. You can go one by one through the folders. You can look in the list here and say, okay, looks like 2, 3, 4 didn't finish, 6 didn't finish, although 6 is right here under 7. I guess there's 4 right there. So again, these aren't all in the exact order, so you'd have to be careful going through and trying to find ones that didn't, and you can be meticulous and do that one by one, or there's a shortcut. If you want the shortcut, there's going to be one command we can run. Now you can come here to the file directory. I'm just going to reload this page. And so I'm within my VBM folder. You want to be in your location. And one last thing before I show you how to do it. If you did the processing and it shows 40 out of 40 jobs completed, all of this you can skip. You don't have to do any of this because you had it finish as expected. I only need to do this because we have some files that didn't process correctly. And so I'll come here, open in terminal, and we'll see if this works. It wasn't working for me earlier. And in that case, yes, not loading. So what I'm going to do is I'm going to use this window. So over on the MATLAB tab. So under Applications, XFCE Terminal, open that. First thing we need to do is get to the location where that Module 7 VBM directory is for you. So in my case, it's here. CD space / blue / sci 4930 / share, and then into my folder. You might be in students and then your folder. You might be in sci 4930 in your folder there, but this is where my folder is. So I have a module 7 and then I have a VBM directory. You can type ls to show we have this con folder and a sz directory. Now over on the GitHub page for this module, we have this command here. You can just copy and paste it, and this will work as long as you run it from that VBM directory. So this will set a variable, the D, short for directory, in either it's going to look in con or sz, and then it's going to look in a subfolder, and it's going to pull out a subject ID from here, and then it's going to look into for a file. Now this command, it will be updated as you run it. I just noticed a little error in that command, so I'm going to fix that really fast. Okay, the command is fixed. The only difference, it was missing a NAT. It has to look in the subdirectory. And so it's going to look in this location for that. So you can copy this file, this path. And so I just use Command C on a Mac, or on Windows, Control C, or you can right click copy. So I copy that, and I'm going to pull it over into here. And you can't just copy and paste directly in this window. You need to go through the side, through this clipboard. So you click on the little arrow, the little tab on the side. The second one down is the clipboard. Paste it in there. And then now that it's in there, I can right click and paste. And what we should see is a list. We are missing subject 2, 3, 10, 11, 18, 19, 56, and some others. So your list, you might not be missing any, or you might be missing more, or kind of fewer than this. So from that list, though, that's what we want to understand is who's missing, because we need to reprocess them. So again, if you had your job complete all 40 out of 40 without any problems, you don't need to do this. If any of them are not completed, then you need to reprocess those. So those are going to be over in the CAT12 window. So this is how we're going to do it. I'm going to click over the CAT12 window. It was still up because I had just left this all up. Click on segment, again like we had done before, and then we're going to do volumes. So this is the double click on here, or you can click on specify. And so now we've got to make sure you're in the correct location, because this defaulted to share data, and that's not where I should be. Where I should be is in share, and then JJ Tanner. Again, my location. And then I'm in module 7, VBM, and then in this location. So there could be faster ways to do it, but I'm just going to kind of take the slightly slower approach. So of course, feel free to skip ahead once you know the process, but I need to go to con, I need to find subject 2. So I'm just going to go con, subject 2, go down, and then I'm going to find their original file. So the nii.gz file, that's the original file. I'm actually going to ignore that one. I'm just going to load in this .nii. So that has been uncompressed. So it's the same file, it's just not zipped, it's not compressed. And so I'm going to click on that one, and that's going to be what I import. And then, so that's subject 2. And so now I want to go to subject 3, who's missing. Again, do the ones that you're missing, if any. So the same process. As we see, within subject 3, it looks different. A little bit different files in there, but here's this .nii. I'm just going to instead of do the compressed file again, I'm just going to do the uncompressed file. That'll save a little step in the processing. Click on it. That's just a single click. You don't want to put both in there. It might say it can't add that other one in there. But again, only do that one. That's this sub file. There are going to be other files created. Very likely, don't put those ones in. That will be a problem. And then single click up. So I'm going to do the process. I'm just going to pause the video at this point and come back once I have them all added. Okay, I have them all added now. You see my list here. And so now that those are in there, I'm going to click done. I have 13 files. Again, that's how many we were missing. And so hit done, 13 files. We don't need to change any defaults. And so now I can just click the green run batch button, and this will take time again. So we'll come back after this is done. Now I'm checking it again, and we see that some of the volumes have finished processing, but only 9 of the 13. That means we have to do that same process again of finding the missing values, these files, those PDF files, and doing them once again. But we can do that by opening the terminal And then I need to get into that location. So the blue Sci 4930, and then share, and then I'm going to where my module 7 data are. Okay, so now I'm in my VBM directory. I can run that same command from before. And this is over on the GitHub page. Paste it in there. And we can see subject 2 and 3 and 69 and 72 did not process. So we do the same step as before. Go back to the CAT12 window and then load the volumes, and we'll find those ones. So I'm going to go into my location, and I'll find subject 2. So there's some more data in here, but the files we're looking for are the ones that are like this sub-02, and then I'm going to do the t1 weighted.nii, and then go up and find the rest of them. So 3 I also needed, and then in the schizophrenia folder I need subject 69 and 72. So we have those added, and then once again can hit run batch, and then when these are done in about 40 minutes or so, we'll be back. So I just want to show that process because it can be multiple steps. For some reason, these files will process just fine for you, but some of you might have to do this multiple times, and kind of this iterative one. Again, so we just want to make sure we know how many files are input, how many files for output. In this case, a total of 40. Okay, we're back, and everything has finished processing. I had clicked around a little bit just to do a couple other things, but I just want to show you, as we look at the MATLAB window, the last 4 that I had done are now finished. They are completed. And so really what we want to do, we'll kind of keep this little streamlined as possible, is I just want to show you a couple of things. So the first thing we're going to do is go into the SPM12 window. So that should still be up at the top. So not the CAT12 for now, but we're going to do SPM12. And we're going to check reg. So check the registration. But for here, we're going to just look at one person's brain. So subject 1, click through, and we have these 5 different files. The MWP1 is going to be gray matter of the brain. MWP2 is going to be white matter of the brain. So we'll select these 5 files and did a shift click there. I'll click done, and it's going to load up the brain. We can see gray matter, we can see white matter, we can see them together, and it's going to just show us, okay, so we have these files, they exist, and we want to see kind of what they look like. And so we could do that for everybody, but really what we can do is say, okay, now we really want to look at everybody's brains together. We want to see if they're all kind of lined up together. And so a quick way to do that would be in CAT12. We can do a check. So instead of checking the registration over here, SPM12, we're going to do this data quality check and do a single slice display. And what we see over here is a place to select the data. So I'm going to double click on that. And now within my VBM directory over here, I'm going to do a caret, so that shift 6, and then I'm going to type in MWP1. And then it should have the dot and the asterisk at the end. Hit the recursive search button here, and it found all 40. So remember, I had to reprocess brains a couple of times, just a subset of them, but now we have all of them done, all 40. And you want to verify that you're having 40 brains at each step. So I select them, I click done, and then I can click the batch. And this will load up all of the scans. And I guess it's not letting me make it larger, but what we can see is just one slice. But really what we're looking for is, does this look like roughly the same slice for everybody? So these are all the different brains. So all are 40 participants. We just want to make sure that we're seeing what looks like about the same structures. So for example, I know it's going to be small, but here, cursor, this is what's called the head of the caudate nucleus. And we see this little separation here between the putamen, this little bit, that dark, that's where white matter would be. That has a little bit of separation. And we can see for pretty much everybody, we have that same structure and a little bit of separation. So the head of the caudate nucleus and this putamen. And that is showing that we have a good registration, or maybe appropriate registration across all of the brains. Okay, so we are a little bit confident at this point that we're looking at about the same place in the brain. So again, if you see on anything that was really different looking on a slice, that would suggest that that brain did not process very well, did not register correctly to the target atlas. We can close that window now. And then there's just one more thing we're going to do. We're going to go back to data quality, and we're going to click on, let me just close that. We're going to check sample homogeneity, and so we're going to do the same thing of select data, and so we'll double click on data to load this up. There we go, sorry. Click on the sample data. And over here on this side, we're going to enter MWP1. Should find all 40 of them. And then we can click the green button. This is going to load data, and it's going to load a plot and some other information. And don't worry about what a lot of this means, but generally what we should have is everything kind of clustered together. We do have some outliers, and that would suggest that there are some potential issues with the data. And but for our purposes, we won't go into that. It starts to be a little bit more advanced. But these individuals, we would check their brains, we would check their scans, and make sure that they're okay, and kind of what's going on in there. But there's different things we can look at quality, and we won't worry so much about that for now. That's a quality step that you would go through. But now we're almost ready to run the VBM analysis. Not quite there yet. But one of the things we would want to do is we need to smooth the data. So I've said in class and other settings that we want to, all the brains have been registered together. They've been all aligned, but that alignment isn't perfect. So we want to do a little bit of blurring. And so we want to smooth the data. So this is going to be on the SPM12 side. And then we'll just come here and click on smooth. And the default, don't worry about quite what these numbers mean, but we're going to keep the default of the 8s in there. So now we want to specify the images, and we're going to do the same MWP1 in here. Search for all these files. So those are the gray matter files, and we will click done. And we can keep all the other defaults in there. And then we will click the green button, and this won't take very long, but I'll stop the video and we'll come back later. Okay, the smoothing is now done. My window showing other things because I was doing some other things on here, but the smoothing is done. We can look at that in different ways. One of the ways we can do is open the CAT12 window, which should be here at the top. So here's SPM12 and the CAT12. And under data quality, single slice display. And what I'm going to do is just look at all of the smooth data. So we can find that from our main VBM directory called SMWP1, and then you should have the dot asterisk there. Click on this REC, we should find 40 files. If you don't find 40 files, then something has gone wrong and you need to go back and kind of process till you have the 40 files. You can make sure you're not going to be processing all of these other ones. So I click done and click on this little green arrow, and it will go and it will load all of these files. So this window will open up and we can see 40 files. They look blurry because they're smooth, and that will just, again, make them a little fuzzier, so that way when we overlay all of them, we are going to be more confident that we're looking at about the same spot in the brain and not catching some of these fine individual variability in the structure of the brain. So that's what now we are ready to use to put in our statistical models. Okay, so the first thing we want to do is set up our model. Now to do this, we will get our total intracranial volume. So that's from the CAT12 window, get total intracranial volume. And we talked about this in class, where it's important in these voxel-based morphometry analyses to include total intracranial volume. And we're going to specify, and we don't need to do anything. So if you're in your VBM directory, then this filter is what is going to be appropriate. You should find 40 files, and these are going to be text files that will have total intracranial volume values in them, and this will save a tiv.txt file. So I click the green button, we can see it collected them, and now we have a new file over here. I can double click and open it, and this file has total intracranial volumes in it. These will be in the correct order as long as you are very consistent in which brain goes first. So I have all of our controls and then all of our schizophrenia brains in that order. So that is our file for that. And now we're about ready to do our group level analysis. So there's different ways to do this, but a simple way is using the CAT12 window. We're going to click on Basic Models. And so now we need a directory, and this is going to be an output directory. And this will be kind of where everything is saved. So what I'm going to do is over here on the MATLAB window, I'm just going to right click and do New Folder. I'm going to call this Second Level. You can call it whatever you want. You could say Second Level Con SZ, kind of schizophrenia, or whatever. I'm just going to keep it simple, Second Level. And that's going to be our output. So now when I select it over here, so let me just show you VBM second level and click on the dot here, and it will show that it's within that location. So that's going to be our output directory. And now we need to start filling in the rest of the things. So to do the group level analysis, we have a factor. Think of that just as our test. And it's not quite our test, but that's how we'll just think about it. We're going to give it a name. You can double click on that or name specify. We're just going to call this group. So our factor is group, meaning our variable. This is going to have two levels because we have two groups. So we have levels and we put a 2 in there. And then under cells, we will need a couple of cells. So you can click new, just a single click, new, And then this cell here, we're going to do levels. So levels is just going to be a 1. Scans, this is going to be all of our controls. So we're going to put SMWP1. Click on the REC button here. We should have 20 files. And then the second cell, levels, we're going to give this a 2. We're going to specify all the scans, in this case, schizophrenia, SMWP1. Should find 20 files and click Done. And then we're going to go scroll down to correction of TIV. If you're following the online tutorial guide, which I recommend, it's going to have, you put this under covariate. We can just do it here under a kind of this built-in TIV here. And so under the vector, I'm going to come back over to the TIV file that I opened up here. Select down all 40 of those. I'm going to right click, copy, and then in this CAT12 batch editor window in the vector, specify, and then I'm going to Control V, or if you have a mouse, middle click the button. And this is going to be true even if you're on a Mac. You do Control V and not Command V. So Control V to paste them in and hit OK. And then we're going to keep everything else there. But now we're not quite ready to run. What I want to do is we want to save this first. So we want to save this in the second level directory, and let's just call it SPM. And then we need to do 2 other things. We need to go to SPM, Stats, Model Estimation, and then here we're going to select the SPM mat file, second level SPM mat, hit Done. Dependency, I'm going to double click on that, and it should basic models spm.mat file. And then one more, we're going to do stats, contrast manager, click on it, select the SPM mat file like before. But now in this case, when we click on dependency, we're going to click on model estimation. And I double click that. And so essentially it's going to do a model estimation, meaning it's going to kind of look overall. And then we're going to do our t-test in this case. So now we need to set up our t-test. So we need 2 new t-contrasts. Those are just single clicks. The first one is going to be con minus sz. Our weights vector is going to be a 1 space -1. So that's going to say controls have more gray matter than schizophrenia patients. The second one, we're going to call this one sz minus con. Our weights vector, specify it. Now it's going to be the opposite of the other one, so -1 space 1. And this is going to be the test that people who have schizophrenia have more gray matter than those who have, or kind of without it, just kind of our control participants. And so this is all set up. Now we are ready to run. So what we're going to, when I click the go button, this will run 2 t-tests, finding areas of the brain where people who have who are controls have more gray matter than people who have schizophrenia, and then also looking in the brain to see if there are places where people who have schizophrenia have more gray matter than controls. And you get an error, hit continue, current directory contains existing SPM file, hit continue because I need to resave over it. You could save it first and it's going to go, and this will be pretty fast to run, as you'll see. We can just keep this live. And then it's going to start popping up different windows, and these are going to be some data that you would look at if you're looking at kind of quality. And we can see we still have a little spinning cursor, so we're just going to wait a little bit for this to finish up. But there's different windows again we can look at, but we really don't have to wait for very long because now it's ready to go. So I can leave these all up, or we can close them. I'm just going to close this window, kind of get that out of the way, but it doesn't really matter. These can just kind of be there. Now we're going to go over to the SPM12 window and click Results. Second level, SPM Mat, done. And now we have a T contrast called con minus SZ. So I'm going to click on that and click Done. And then apply masking, none. And then do we want to adjust the p-value? Now, we only have 20 people in each group, but let's see what happens if we do some robust statistics. So if you're going to do this for a real research project, you might skip a p-value adjustment first, but to publish it, probably going to do a family-wise error correction, or you don't do an adjustment and you could do some sort of adjustment after the fact. That's more advanced. We won't go into that. But I'm just, for my own interest, I'm going to click on family-wise error adjustment. I'm going to keep the 0.05. And then I'm going to put 50 voxels in here. Meaning we're going to look for, it needs to be a pretty good little cluster of voxels of difference. And I hit enter return, and this window pops up, and we see one little black spot here. So what is this? This is actually where people who have schizophrenia have less gray matter than our kind of control participants. And this is quite a robust finding, actually, because we did this family-wise error correction. We only have 20 in each group. This isn't a huge sample, and this is showing this area. Now, you could submit a screenshot of this for the homework, or you could do one more step. I can go to the overlays here, and then I could go to sections, and then in here, we need to go to this location. We're going to go to blue Sci 4930, share NeuroTools. Sorry, data NeuroTools / NeuroTools SPM12. And then within canonical, there's this single subject T1. Click on that single click, hit done. And what this is going to do is we'll load up a brain. And if we scroll up, we're going to find this area, just one small kind of spot, but right here. So this is an area of the brain, kind of roughly anterior cingulate gyrus, a little bit of kind of the bottom part of inferior, kind of this inferior part of what we call superior temporal gyrus, but in this kind of, in that region near the anterior cingulate cortex. And this is an area that's doing a lot of kind of emotional regulation processing and some other things, but we're seeing this difference. A very robust statistics there. So you could do a screenshot of this. There are different ways you can kind of export this, print this, and kind of save it as a PDF or something and submit that. But a screenshot of this would work for the homework. Now, let's just look at the other contrast, meaning results, load this matrix file again, the map file, and then let's look at schizophrenia minus control. So this is going to be, do people with schizophrenia have more gray matter than controls? And click none. We'll do the same family-wise error correction, and it's going to load, and we don't see anywhere in the brain that shows a difference. So that's showing people with schizophrenia do not have more gray matter than our control participants. Now, just for interest, I'm going to be a little bit more generous with thresholding. So I'm going to go back to our control, minus schizophrenia. I'll do none there. We won't do a p-value adjustment, but I'm going to make this less than 0.0001. And then I'll still put 50 voxels in there. So if we look, there's quite a few spots in here. And so this gives you a whole output, a table. This table could actually be like output, almost ready for a kind of publication, for a submission. And go back to the overlays on this section. We would need to get back to that location. So share data, NeuroTools, and go into the SPM12 folder, canonical. You could load any of these, but we'll load the single subject one. And now we can see a few areas of the brain. So again, that same kind of anterior cingulate, a little bit of superior frontal gyrus. We can see some other areas where people who have schizophrenia have less gray matter than our control participants. And so they're kind of all over the brain, temporal lobe over here. And we're kind of getting, but a lot of frontal, some temporal, and a little bit in our kind of posterior temporal, occipital cortex, and kind of these different regions. And so the question is, well, do these relate with behavior? And so those can be some of the next analyses that you might do with a research study. But again, this will just show if there's some differences. Now, these data have been published already. These are publicly available, meaning these analyses have been done. But I mean, this isn't particularly new data, but if you were doing this with a novel data set, you could see some findings and publish this. So let's just do one last thing. And you don't have to do this. I'm just showing you if you want to be a little bit more generous, kind of liberal in the stats, then we can, I'm just going to put one more 0 in there. And again, we notice that even with more generous stats on that, we don't have anywhere in the brain where people with schizophrenia have more gray matter than controls. So that should tell us at least a pattern that something is happening. So schizophrenia, again, is very neurological and kind of neurodegenerative, that there are these areas of the brain that there's less gray matter and something's happening. And we know that people with schizophrenia tend to develop more cognitive impairment, memory problems, and some other conditions. And we can kind of get a sense of that something in the brain is causing the loss of gray matter. Now, these are cross-sectional data, so I'm making some inferences there, but again, this longitudinally, some people have shown that there's this loss of gray matter and changes in the white matter and kind of a lot of changes throughout the brain in schizophrenia. So again, you can submit things, but I just want to give you the walkthrough, and that's the module.